I'm zooming in from upstate New York, and my lab studies the molecular mechanisms by which the brain constructs itself during embryonic development. And one of the things we're really interested in is understanding the really important role that POGZ plays in this process of brain development. Um, so, so POGZ was actually first identified as a gene linked to autism from these sequencing studies that were conducted almost more than 10 years ago. And now geneticists have analyzed more than 20,000 individuals with an autism diagnosis, and POGZ, sorry, comes up as one of the top genes in these analyses. And so it is clear that POGZ is very important for brain development. And also POGZ is linked to white Sutton syndrome. And so heterozygous loss of function mutations are known to cause several of these phenotypes, including intellectual disability, language delays, autism, and other co-occurring conditions that I'm sure you have all been talking a lot about in this meeting. And so I was very curious to understand what POGZ does in the developing brain to try to understand how, how we can potentially find a therapy for this disorder. Because very little was known about POGZ when we first discovered this gene, when scientists first discovered this gene around 10 years ago. Nobody had ever heard of POGZ before, and very little was known about what it actually does. And so what our work is showing and others have found is that POGZ is an epigenetic regulator. So what is epigenetics? So genetics refers to the DNA within our cell's nucleus. Epigenetics is everything else that embellishes the DNA within the cell's nucleus. So as you know, the cell's nucleus is like a bowl of spaghetti where the DNA is wrapped around these balls of which organize the DNA into silent and active regions. So the silent regions are tightly compacted, and you can see the DNA here in black is not very accessible, is not available, while the DNA in the active parts, there it is available to be bound by other factors and to be functional. And so epigenetics refers to how DNA is packaged within the cell's nucleus. And this is a very dynamic process during development because at different stages of the developing brain, different genes are important and need to be active, and other genes are not so important and need to be silenced. And so we believe that POGZ controls this balance and this regulation within the cell's nucleus between active and silent parts of the DNA. And so what we have been doing for several years now is analyzing this mouse model that we have made, where we deleted POGZ using CRISPR-Cas9, and we deleted a large portion of this gene between exons 1 and 7 in the mouse, and this led to a homozygous loss of function. The homozygous, so when you lose the gene on both copies, on the mom's and the dad's chromosomes, this is embryonic lethal. So the mice don't survive after birth. But when the mice are heterozygous, which means that they're missing only one copy of the genes, which is the situation that most resembles the human phenotype and the human genetics. These mice do live, and they had some interesting behaviors that we were able to observe. So, for example, they have reduced anxiety, actually. So we put them in this, it's called a maze. It's more like a plus sign. And the mice prefer to spend time in these closed arms of the maze rather than these open arms, which are like a scary balcony they're walking on. And so what we found is that the POGZ heterozygous mice spend more time in the open arms in the scary parts of this maze than the other ones. And so this indicated that, and they actually like peek their heads over the sides of the edge. So these mice seem to have differences in their behaviors in this really well studied anxiety-like behavior in mice. And actually, what we saw is that as the mice approach the center and they have to make a decision, sorry, do I walk in the open part or do I stay in the closed part? The POGZ heterozygous mice, usually two brain regions start to talk more to each other as the mice are prepared to make this decision. And this increased communication between two different brain regions is indicated by this rise here in the teal line, while the POGZ heterozygote mice did not have this increased communication. And so we think that in these mice models that we've generated that there are deficits in the communication between the prefrontal cortex and the hippocampus, and that this might be leading to this behavior that we observed. Okay, so I mentioned that we have observed something interesting in the heterozygous mice. But we're primarily focused on the homozygous mice where POGZ expression is completely lost. And so what this mouse helps us with is to understand what does POGZ actually do in the brain when it's really highly expressed. So here you're looking at a section of a mouse's brain, so like a coronal section, like if if the brain has been sliced where the crown is placed. And so you can see here is a section of of a mouse embryonic brain. And you can see that POGZ is very highly expressed in red and in purple are the protein levels and the RNA levels of POGZ. And then in the knockout, we don't see red and we don't see purple. And so this is indicating that we have generated a mouse where there is no POGZ in the developing brain. And so then we look to see what other genes are affected in this mouse, which is completely lacking POGZ during its brain development. And we saw that some other genes that are very important for the wiring up of the mouse brain are also affected. So we see these genes that have, so SLITRAC1 and SLITRAC5 have actually been implicated in Tourette's disorder. And we're seeing that these genes are also downregulated, are not expressed as highly as they should be in the POGZ knockout. Okay. So then we're very interested in like what does POGZ actually do within the cell's nucleus? How does it control the DNA? How does it control which genes get expressed? And so we're doing these experiments. This is like biochemistry to map where in the genome is POGZ binding. And so we're finding that POGZ does bind DNA. This map shows us, and these blue bars indicate locations in the genome where POGZ binds. And in the knockout, we do not see any blue. So this is a good control. Shows us that our experiment was well done to show that POGZ binds DNA at around 2,000 locations in the mouse's genome in the developing brain. And so what we think our analysis is showing is that POGZ binds DNA sequences and controls the expression of other genes that are very important for neurons to grow and form connections. So this is a summary of what we've discovered about what is POGZ's role within the cell. And how does it contribute to the development of the brain in mice? And so now in my lab, we're continuing to try to understand what are the parts of the genome. So within the bowl of spaghetti that is the cell's nucleus, which specific loops of the spaghetti does POGZ control? And so does POGZ organize the DNA within the nucleus? And so we've been again focusing, analyzing these knockout mice. So here again is a cross section of the mouse embryo brain, and we dissect out the different brain regions and do this technique that allows us to map the DNA within the bowl of spaghetti of these cells. And so what we see is like these red pyramids indicate one loop of the DNA. And so the DNA is organized within different loops within the cell's nucleus. And we're finding that POGZ is only responsible for organizing the DNA of a subset of the genome. So POGZ isn't important for the whole genome, it's just really important for a subset of the genome and just a few genes, a handful of genes. And these happen to be genes that are important for neurons and for the brain to develop normally. So this is another example. We see these pyramids indicate an organization that is dependent on POGZ because in the knockout we see the pyramids disappearing into the mist. Okay. So we are starting to understand and to appreciate that POGZ plays a very important role in the developing brain. It's important for not for the whole genome, not for every single gene in the genome, but just for a subset of the genes in the genome. And so we also are starting to appreciate that one other gene that is closely linked and that does something similar to POGZ is called ADNP. And kids with mutations in ADNP also have a similar a set of phenotypes to the White Sutton syndrome kids. And so we were surprised to do molecular studies and to find that POGZ and ADNP are found together within the cell's nucleus. So they're interacting and they regulate the same parts of the genome. So this is very striking and important to show that POGZ has a partner in crime that is ADNP and that they both do the same kind of thing in the developing brain to regulate the same subset of genes. And actually, it seems that ADNP is itself regulated by POGZ. So here we have a mouse's brain, and you see that when you're lacking half the amount of POGZ, you also lose half the amount of ADNP. And so we do think that these two genes are really tightly linked, and they might be controlling each other as well. Okay, so our paths forward for POGZ is to identify which are some common genes that depend on both POGZ and ADNP. This will help us really whittle down and figure out what are the downstream targets of POGZ that we should be focusing on. Because the fact that ADNP is also there regulating the same genes is a good sign that these are implicated in the pathology. We are trying to identify which cells in the brain are deficient in these genes that POGZ controls. And then ideally, we would like to be able to boost those cells and their connections in the brain. Because they were missing these other genes that I talked about, as a result of the POGZ mutations, we want to be able to consider different approaches to boost those cells' connections within the brain. And so we're aiming to do these experiments in the mice. And finally, we want to model actual patient mutations. And we're doing this in mouse cells and in human cells. So we have made a transgenically modified mouse where we introduced a mutation in in one location in the gene POGZ, in the mouse's version of POGZ. And so this is a mutation from a child with a severe form of White Sutton syndrome. And and so we were able to use CRISPR gene editing approaches to introduce the mutation from this White Sutton syndrome child into the POGZ gene. And then we're really interested to see, so now with these mice, the heterozygote mice, they're fine. And so what we're noticing here in this experiment is we're looking at the levels of POGZ, which is this dark band, oops, in wild type, and then in knockout and in heterozygous. And what we're seeing is that this mutation leads to half the amount of POGZ being made. So what we weren't sure what to expect is does this mutation in this location of the final exon of POGZ, does it lead to a shorter POGZ being made, or does it lead to a same length of POGZ being made but with smaller amounts of the protein? And it's actually the second case, that the full length, it's the same size, which is the height on this graph of in the mutant, but it's just not as blazing hot as it is in the wild type brain. So we are now working closely with Simon's Searchlight. So the Simon's team is collecting blood samples, and they were there in Orlando last year also. And so we are now analyzing the cells from the blood samples that were collected in Orlando. And we're trying to repeat these experiments to see whether the different mutations lead to half the amount of POGZ being made and or to a truncated protein, a shorter protein that is unable to function as the wild type version of the protein would. And so I'm very grateful to everyone who contributed to our research through these donations. And I want to thank everybody in my lab who participated in this work. Specifically, Natasha has been leading the work. And also, I'm very grateful for the collaboration with Simon's Searchlight. And I'm happy to take any questions.
Thank you so much. This is amazing, and we are appreciative that you are sharing this information with us.
Of course.
We have new people signing up to give blood samples, and Simon's will be coming to our door this time since they couldn't make it to our conference. So we have a good list of that, which is exciting for
That's
further
great.
research for you as well.
Yeah.
Your science is fascinating, and we appreciate you updating us. Any questions? Yep, hang on just a second.
Yesterday when Astrid was talking, she mentioned that you're running out of the samples. Is there a way to have an automatic, a text or an email set up to the families that you need a redraw?
Yes, I could definitely help with that. And I will work with Amanda to get that letter out. And yeah,
Thank you. Yeah.
because the all the paperwork is quite is quite intensive, and we only recently got the cells from Orlando. It would be great if the samples are sent through and the samples are collected with the Simon Searchlight team.
Okay.
Thank you for that great talk. I had a question about, you mentioned that there's some abnormalities and connections between the prefrontal cortex and the hippocampus. Can you elaborate a little bit more on that?
So yeah, so we these were from experiments where we put these probes in the mouse's these poor live mice had these probes in their prefrontal cortex and their hippocampus as they were walking around the maze. And usually you see these like oscillations, these vibrations in the brain, just even a sleeping animal will have these oscillations, but especially as the animal is walking around and making decisions. Usually the oscillations in these two brain regions sync up and they kind of like go in sync with each other as the mouse is preparing to make a decision, like do I turn left, do I turn right? And what we're seeing is that there is no such syncing up of these theta oscillations, they're called. So this is kind of a really striking finding, and like so these are very well studied communications between these two brain regions that are really important for these anxiety related tasks.
Have you, just a follow up, have you
Hmm.
analyzed like brain sections to see if there's any
Yeah,
trajectory abnormalities?
yeah. So we really want to do that to see like are the neurons actually not able to connect in the right way between these two brain regions. That's a great idea.
Hello, thank you so much. I'm so fascinated by everything you're sharing. One of the
Hmm.
slides was showing variant samples from patients' blood that was collected in Orlando last year.
Hmm.
It appears that my daughter, for example, might be one of the ones that it says was not shown due to missense missense
Hmm.
variants. I was wondering if you might be able to explain what that is or what that might mean.
Variance, VUS stands for variance of uncertain significance. So is if it's hard to predict what effect a mutation will have on the protein's function. So within these other variants here that have like a little asterisk indicates that the mutation introduced a stop. And so the gene just will stop being made. And so even this is not indicative. You need to actually experimentally test the effect that a mutation will have on the protein levels and on the protein size. It's really hard to predict how any mutation will affect a gene's function. And so the two Simons searchlight variants of uncertain significance, VUS, just means that it's even harder to predict what those mutations and how they will affect the gene because they're not introducing a stop, but they're changing one amino acid to another. And so this is exactly a case where we need to be able to. So here they are at the top, the two variants of uncertain significance. And so it's very important, especially for these ones, to experimentally test the effect that they have on POGZ. It could be affecting how it interacts with other genes like ADNP or how it interacts with the DNA.
All right, thank you so much. Yes,
Hmm.
I'd be curious to learn more.
Yeah, me too.
Anybody else? No. Thank you again so very
Yeah.
much for
Thank you, Amanda.
educating us on your science, and
Hmm.
thank you for doing what you do.
Of course.
We appreciate you and your team.
Yeah. Thank you. Enjoy the rest of your meeting.
All right. Bye-bye.
Bye-bye.